And it's a great privilege to be introduced by somebody whose work you refer in your lecture. Also would like to thank Duncan and Sofian to give me this opportunity to talk about what small things we are after, especially at this time of the day when the food for our thought should be none other than literally the food that we're going to have for lunch. So it looks like Tesco is having a center stage, and our department unfortunately is not anywhere angstrom distant from Tesco, but it looks like the place I live in, Europe's biggest Tesco, is there. So I should be an honorary member of the club. Anyway, so as a lab we are quite thinly spread, and we are after various things. Everything is little and probably underwhelming. So today we would like to talk about something that I had an opportunity to talk about last year on the CRAC channels. So just to have a really brief catch-up, the CRAC channels underlie the so-called store-operated calcium entry, meaning every time the internal calcium store like the ER or SR gets emptied physiologically by messengers like IP3 or pharmacologically by a sarcopamine inhibitor like tepsigargin, then following the rapid release from the store, there is a sustained entry happening in the plasma membrane, the so-called store-operated calcium entry, SOC, SOC, whatever you call it. And it's really lend itself to very easy experimental capturing by simple calcium imaging. We can live with calcium-free media, apply tepsigargin to deplete the store, and we can put the calcium back. We get a rapid influx, and in this way you can segregate both the release and the entry from each other. However, electrophysiologically, it's probably a nightmare. Not any student is very keen to record this channel because the problem comes from the tininess. This is a slide I stole from one of the Sofian's application note. It looks like they had a QPatch protocol optimized for iCRAC. But yeah, it's inwardly rectifying, and I think based on the literature, it is the next to L-type calcium channel in terms of calcium selectivity. It's very highly calcium permeable. And in my opinion, this is, or everybody agrees that most of the cases, in most places in the body, the CRAC channels are the in situ protein-protein interaction mediated channel complex made of two proteins residing in two different membranes, the ori present in the plasma membrane, steam present in the membrane of the ER. When the store depletes, they cluster and they migrate to each other, and they aggregate to the point where the ER and the PM are in a close apposition. And together, these form this channel. So this weirdity makes it interesting. And a lot of great scientists working in these channels. I'm no longer in that club. However, and it looks like initially it was thought to be a feature of the immune cell, but it's everywhere, including the excitable cell like heart or neuron or skeletal muscle. And it makes sense because every time we release calcium from the store, we need to refill the store. So one of the long-standing role of this pathway is to store refilling. But given the fact that the calcium is not very readily diffusible, it kind of creates a calcium cloud, wherever it is generated from. And unsurprisingly, SOC or CRAC-mediated calcium signaling has been implicated to various specific cellular roles. A biggest role comes from the immune cell, where it was first identified. It looks like this calcium calmodulin that is activated following this calcium, specifically coming from this pathway in T lymphocyte, can actually activate calcineurin, the phosphatase, which dephosphorylate the highly phosphorylated NFAT, which then move to the nucleus. And you can track this by simple confocal microscopy, where you can tag the NFAT. This is the resting field store. You can see mostly cytosolic, a few in the nucleus. Whereas if you apply tepsigargin or ionomycin, you can see within probably a few seconds or maximum 5 seconds, it does go to migrate to the nucleus. And actually, many molecules against this pathway was identified by this phenotypic screening. So after going to the nucleus, obviously NFAT then upregulate transcriptionally various pro-inflammatory cytokines. Many of them are also important for immune cell proliferation and function. So a lot of disease are associated with hyperactive CRAC channels that leads to hyperactive immune cell, leading to all sort of autoimmune disease and then ARDS. There are also immune equivalent cells in the brain, the microglia. The overactive CRAC also leads to hyperactive microglia or microglial polarization, feeding forward to neuroinflammation and neuropathic pain. And also there is a calcium toxicity caused by this source, and that is where mostly some molecules are in being clinical trials, like pancreatitis, kidney injury, stroke, neurodegenerative diseases. And there were some early companies who had some molecules. I don't know why they lost interest. By now there may be a reason for it, but there are also some new generation companies who are actually taking it really forward in various cases. So there are a lot of molecules now, and the universe, the chemical space is expanding. Some of them are reasonably highly selective, some of them low selective, and there are non-selective all sort of molecules. This is like some prominent ones. So you may ask actually, do I actually dare into this jungle? We did dare actually. We had some ligand-based, similarity-based finding and found some molecules that have an effect on this pathway. So we also now, we means the whole community have got Drosophila CRAC channel structure solved in both closed and open state. This is a closed state. So we thought, how about we do give it a go, the structure guided. The main reason is all the molecules that we see, excluding barring this approved drug for other things, they're all phenotypically obtained. There was no structure information, no target information, and a lot of them originate from one molecule called BTP2. So the only literature we are aware of that was done in a very rational way and they knew how their molecule work is this paper from Park Group, where they actually took some minimal domain from ORI and fused to GST fusion protein and did some small molecule microarray. To cut a long story short, they got this kind of flavonoidal kind of scaffold that has around lower, like less than 1 micromolar potency. And they did model where the molecule could be binding to is a region of the ORI that interacts with the steam molecules. But beyond this, another thing that disturbed me as a pharmacologist is that all of molecules, nobody is open about how they work, but they are in clinical trials. And the other thing is when we have students, we struggle to reproduce especially on the potency side of things, a lot of the molecules we buy, in the literature, people treat them from 10 minutes to 1 hour to 6 hours to a day to 48 hours or even 72 hours, ranging from nanomolar to high micromolar to see some significant inhibition of this so-called CRAC pathway. So it was a bit puzzling. Anyway, how did we start? We started with these 4 molecules that look kind of similar because they all originate from this father pyrazole compound called BTP2. And I think what we did, we wanted to know actually which side of the membrane they work. Is it from the extracellular side or the intracellular side? So we did a wholesale recording, not the easiest one. And you can see the BTP2 only worked externally when it was included in the pipette. In the blue line, it didn't inhibit the peak current. PYR6, which is so far the most selective one that we can buy off the shelf from the vendors, it seemed to work both externally or internally. We don't know whether the externally means it could readily permeate or it hits on an additional binding side. The GSK compound, one of the early CRAC channel blockers, it also mimics kind of like BTP2, only work externally, it didn't work internally. The Cinta again goes back to the PUR6 kind of phenotype. It can work both externally and internally. So it was in a way better than having, groping in the dark, that at least relative to the side of the membrane, which molecules work potentially in which side. The next thing we tried, the obvious thing, resorted to docking software to predict some sort of binding poses. The thing is, if we just analyze CRAC channel as a protein and ask some algorithm, can we find some druggable hotspot? It never picks up any hotspot from the extracellular domain, whereas there are quite a few hotspots from the cytosolic end. So that's why we went to blind docking and we found one poses where molecules seem to cluster quite often between these two pore-forming helices. This structure of the CRAC channel, these are basically hexamer. You can think about like a trimer of a homodimer. It's a very weird orientation. And this inner circle of helices are the M1 helices that face the pore of the channel. And then the intercellular pocket was analogous to what that paper was reported to. We focus on the extracellular one for the time being. So this thing we did, we have a student like who joined us in the audience. We just did a 100 nanosecond atomistic simulation, putting these big things in a membrane, and ran 100 nanosecond sometime in triplicates, in few cases even 5 times, to see whether the poses, the molecule stays there or not. So these are the examples where you can see it hasn't left the place. So we just took it as a prediction. Okay, this could be a plausible place or happier place to keep the molecules there. One caveat is this: we use, based on the what simulation people have done, a normal sodium potassium solution. We didn't have any high calcium. So one of my hypotheses, maybe the permeating calcium may also engage with this blocker, as with the case with some calcium channels. But this is a future to explore. The mechanism could be either full or partial occlusion of the pore. So we did the obvious thing, focusing on this pocket, did some screening with some diversity library from Enamine, from Cambridge or SPEC, and used this normal calcium imaging assay. We can look at both the peak or the steepness of the rising phase, which indicates the rate, or sometimes people also measure the area under the curve. In various ways, you can get some idea about how much calcium is feeding through this CRAC pathway or not. And then for the interesting one, we feel incentivized enough to go for the iCRAC recording. And then the round of repetition, you can test for selectivity against other calcium release pathway and entry pathway like voltage-gated or TIFC, NFAT translocation to the nucleus assay. Again, if we had money, go for ELISA-based chemokine secretion, concentration response stuff with the good, interesting looking molecules. And then until we are happy, we can, and until it is feasible, we can keep repeating the cycle of design, make, and test as implementable as in an academic lab. So this is unfortunately a blood one. I didn't mind because we're not after like a gold, a highly super holy grail, but my students said, can we make it blood? I mean, okay, we will do bloodify whatever molecules. So we bought 50, we shortlisted 50 molecules. We bought 27, we tested them. 15 were active at 10, 4 were active at less than 5. Industry people say, oh, these are like not really good hits because it's a very still micromolar. But for us, and also from my personal point of view, is I wouldn't mind having a low micromolar acting molecule as long as the selectivity is good enough. Potency can be improved after med-chem optimization. So our best molecule is, or CRAC, I call it, CRAC inhibitor, is this compound, E1, which has got a potency of around 5-ish micromolarish in the calcium assay. Its structure does look novel. It prevents NFAT translocation. You can see this is the HeLa cell expressing the human NFAT tag with GFP. This is with tepsigargan. You can see all in the nucleus, or a lot of in the nucleus, whereas in presence of this molecule in 5 micromolar, there's significant absence in the nuclear greenness. It's not cytotoxic up until 50 micromolar, which is a good sign. It does reduce interleukin-2 production in an ELISA-based assay, with seemingly greater potency than what we see in the calcium imaging. And it was reasonably selective, at least against some pathways we could test, like IP3 calcium release, then ryanodine receptor caffeine calcium release, then TRPC mediated calcium entry, then also L-type channels using some excitable cell line like InCell. They remain active, at least at a concentration which they can still inhibit the SOC. So the other thing we try to do, like all the previous, these experiments where we incubate the compound at least 10 or 15 minutes before we take it to the imaging. So we thought, okay, let us try a more acute challenge, like just before adding the calcium, let's add the compound and then add the calcium as quickly as we can. Idea is if the molecule is acting extracellularly or blocking the pore, they will have an effect unless the molecule is so rapidly permeating, which is unbelievable. So anyway, it does look like the molecule, you can see in a concentration-dependent manner, it does very rapidly inhibit the calcium influx just before adding the calcium in the assay. So it kind of aligns with the EFIS data as well. We did the same thing, put it in the bath, put it in the pipette, and see in what way. You may tell that you may not have left it in the bath for long enough to allow permeation and all these. We tried if we can maintain the cell for maximum up to half an hour, not always, in many cases, but still there was no kind of sign, especially, and here is more redundant because here the molecule is active in the bath, and if you leave in the pipette, and there was a referee asking us, what about your molecule is effluxing out through passively? So that's why we actually did some experiment in the E-phase and lived in the pipette for half an hour, but we didn't see any inhibition. Anyway, that was reassuring. At least we have a dog that is wagging the tail for a good reason, because we found it in that way. And then unsurprisingly, when we tested them against their ability to whether it interacted or messed up with the interaction of steam with ori or interaction between steam or between ori, these are some Hexel liner I requested from a US-based researcher, I forgot his name, which I should remember, the M-Cheritac steam and the GFP-Tac ori. This is the normal control cell where the store is filled, and when we have CRAC, you can see the punctate distribution of the ori, and also punctate distribution, I don't know whether it's visible or not, with the steam, and you can see some yellow spot, meaning they are also interacting or talking to each other. And then in presence of this compound, 3 micromolar, for example, you can see there is no obvious problem in this kind of punctate distribution. We did quantify, but it's not present here. So, however, as I said, when we did the simulation for some of the molecule, we did simulation 5 times, 6 times. There was a reason for it. One is this BTP2. You can see, sorry for this movie going on beyond my control, but you can see they start from here, the BTP2, which is similar to between the helices, and then it kind of jumps around and then kind of becomes happier somewhere beneath this M1. The GSK compound also did the same way. It kind of moves soon after the start of the simulation. It flew back to the other place, which is initially equivalent pose, and then it settles behind these helices. So we could have disregarded the observation, but we thought maybe this is another pocket that we couldn't spot through docking. So this is another place we are focusing on. If we then redo the same cycle of molecule finding and get some molecule working in a way they should, it probably could validate this pocket as a potential druggable pocket. And if it was a real pocket, I think the idea is probably it kind of allosterically distort the geometry of the pore, or maybe partially wedging into the pore helices. Either way, the calcium will have a problem to pass through. So I think at the end of the day, if we ask about the achievement, I would say that we could significantly reduce the haystack size, because we only bought few molecules, and I think that's where, as an academic lab, I'm always interested. Can you buy less, still get some hit? Okay, many people also do it the same way. We also kind of address what Socrates would have told us if we were alive, then know thy molecule. And I would like to end by returning back to TESCO. I think our work also reproved that every little probably helps. So thank you for your attention, and I'll be happy to receive questions.
Thank you very much indeed for this very stimulating talk. Are there any questions? Yep, over there.
So maybe this is out of focus and I'm not into this at all. But yesterday we heard about cilia and at least as I understand it in the field I'm in, the beta cells and the cilia there, they have calcium-induced calcium release. Do you know anything if CRAC channels are expressed in cilia?
I would be surprised if they are not, because if the cell wants to reuse the store obtained calcium, it has to refill it. Although having said that, excitable cell like neurons, they does it through the L-type channels, so they are not obliged. But I don't think anybody has looked at it. It may be my non-comprehensive scanning of the literature, but I would be surprised if they are not there, to be honest. So much ubiquitous. Some people even still are very skeptical about how good it can be a target given the ubiquity of the expression of these channels.
Any more questions? I had a question about, you were looking at ORI1 there, were you?
This is the wild type, but this is the work in progress. Now we've got cells, again, brought from Mohamed Tribag, ORI1, 2, and 3.
Okay.
So we're testing them sequentially, which one is the one that probably has, is there any selectivity that we may be lucky to find?
You have no data on that?
Not yet, it's the work in progress at the moment, yeah.
Okay, thank you very much.
Thank you.
Thanks a lot.
Oh, thank you.
Thanks a lot to all the speakers.
That's all right.